Hi everyone, I'm Erva. I'm delighted to have this opportunity to present my project, Mutual Annotation-Based Prediction of Protein Domain Functions with Domain2Go. In this presentation, I will walk you through the methodology we developed that leverages mutual annotation to enhance the accuracy of protein domain function predictions. To begin, let's dive right in with some foundational knowledge about protein function prediction. Understanding unknown protein functions is vital for unraveling biological processes. However, traditional wet lab experiences are time-consuming and costly, and to overcome this, computational strategies have been developed to define and identify biomolecular functions. These strategies rely on transferring annotations between proteins with shared characteristics like sequence, structure, interaction, or profiles. It's crucial to establish a systematic and machine-readable approach for annotating biomolecular functions, and biological ontologies like gene ontology offer standardized vocabularies for this. Establishing functional associations, especially for complex and multifunctional proteins, can be challenging. To address this, we can leverage the modularity of proteins. Domains acting both as structural and functional units influence protein behavior at the molecular level. Analyzing a protein's domain composition provides valuable insights into their functioning within complex biological systems. In this research, we introduced an innovative approach called Domain2Go, which aims to identify previously unknown protein functions by linking their domains with gene ontology terms. To establish robust domain Go term associations, we examined the co-occurrence patterns of domains and Go terms on the same proteins, employing statistical resampling techniques to ensure reliability. We then utilized the train and test datasets from the critical assessment of function annotation to evaluate the effectiveness of these highly reliable domain Go mappings in predicting protein functions. We compare Domain2Go in this challenge against other existing methods to assess its performance. As a result of our study, we present Domain2Go as a programmatic tool readily available for generating novel protein and domain function associations. By providing this tool, we aim to enable other researchers in the field to leverage and benefit from our approach, facilitating further advancements in protein function prediction and annotation. In this figure, we can see the overall methodology of Domain2Go. This approach generates domain Go term mappings by combining domain information from proteins with their corresponding Go term annotations obtained from Interpro and UniProt gene ontology annotation databases. To ensure highly reliable associations, we utilized statistical resampling techniques and analyzed the co-occurrence terms of domains and Go terms on the same proteins. We calculated three distinct probabilistic association measures using the expectation maximization algorithm. These measures allows us to assess the significance of Domain2Go mappings and evaluate the predictive performance of our method in an ablation setting. The finalized domain Go mappings were generated by applying a threshold to the association scores. Finally, we applied Domain2Go to predict currently unknown functions of proteins in the UniProt KB SwissProt database by propagating domain-associated Go terms to full proteins containing those domains. To generate domain function mappings, we combined two datasets. For the domain protein dataset, we obtained the UniProt KB Interpro entry annotation file from the Interpro database. As for the protein function dataset, we downloaded Go annotations with manual and experimental evidence codes from the UniProt Go database. To enrich the annotations, we propagated the Go terms in the dataset to their parent terms based on their ISA and part-of relationships. Next, we continued by mapping these two datasets by utilizing shared UniProt IDs between domains and Go terms. When a UniProt ID was found to be co-annotated with both a specific domain and a Go term, we considered this pair as a raw Domain2Go mapping. To ensure the reliability of the mappings and eliminate any coincidental occurrences of a domain and Go ID on the same protein, we performed a filtering operation. This step aimed to refine the mappings and enhance the accuracy of the Domain2Go associations. For this, we calculated a co-occurrence similarity score for each raw Domain2Go mapping. The score was computed using this equation here. This considers the number of proteins co-annotated with a specific domain and the Go term, as well as the total number number of proteins annotated with this domain and the Go term individually. Here we can see an example of the co-occurrence similarity calculation on a toy dataset. Higher co-occurrence similarity score indicated that the number of proteins co-annotated with domain I and Go term J was significantly higher compared to the total number of proteins annotated with these terms individually. This score served as a threshold to differentiate relevant domain-to-Go pairs from chance occurrences. To account for the possibility of high similarity scores being random, especially when the N, DI, and N, Go, J were low, we introduced an additional measure called lowercase n, representing the number of proteins with co-occurring annotations. Domain-to-Go pairs with both high S and N values were deemed more reliable. To determine optimal thresholds, meaning S and N values, we compared the co-occurrence similarity distribution from the domain-to-Go mappings with a distribution generated from randomly paired domain-Go terms. By examining the differences in the frequencies of high and low S values between the original and random mapping sets, we established suitable thresholds. To create the random mapping set, we shuffled the domain and Go term columns in the original mappings as we can see here. Subsequently, we recalculated S and N values for the newly generated random mapping. We then generated histograms of the co-occurrence similarity distributions by applying selected combinations of S and N scores as thresholds to the mappings. We utilized the Kolmogorov-Smirnov test, a commonly used non-parametric test to compare two samples and determine if they are drawn from the same distribution. This test quantifies the distance between the samples and evaluates whether the null hypothesis, stating that the samples are drawn from the same distribution, is true. By applying the chaos test to the original and random distribution pairs we generated, we aimed to identify the minimum combination of S and N values that demonstrates a significant difference between the distribution of the original domain-to-Go mappings and the random associations. This minimum S and N value pair was selected as the official threshold. Here we can see the statistics of the finalized domain-to-Go mapping sets after the thresholding operation. To evaluate the prediction capabilities of different statistical association scores for domain function mappings, we utilized three main association scores calculated by the expectation maximization algorithm. The first score, abbreviated as theta-E, provides an initial estimate of the probability of association between a domain and a Go term. Through multiple iterations, the EM algorithm refines this initial estimate by calculating various submeasures. These submeasures include factors like number of proteins co-annotated with domain E and Go term J. The second main score, denoted as theta, is the refined version of the initial theta value. This is obtained when the likely score reaches a point where it no longer noticeably increases during the iterative EM process. The third main association score is referred to as E. Unlike theta, which represents the probability of a mapping, the E score aims to identify highly specific domain-to-Go mappings supported by substantial evidence in the co-annotation data. To calculate this score, we rerun the EM algorithm under the assumption that the probability of association between a domain and the Go term is 0. A lower E score indicates that a pair is not significantly better at explaining co-annotation compared to other potential domain function pairs that share the same protein annotations. To evaluate the accuracy of different association scores, we conducted an enrichment analysis. We ranked the predictions for each of the four measures and assessed the number of ground truth domain Go term pairs that appeared within the top K predictions. The ground truth pairs were derived from manually created interpro-to-Go dataset. To estimate the expected number of ground truth mappings in the top K predictions, we used the ratio of interpro-to-Go verified pairs across the entire prediction list. The enrichment plots show the performance of each association score. Notably, the S score outperformed all other measures in identifying significant domain Go term pairs, particularly at the low rank cutoffs. Based on the findings from the evaluation study, we decided to proceed with the mappings obtained through co-occurrence similarity-based filtering. We compared our finalized domain-to-Go mappings with interpro-to-Go, a manually curated domain function annotation dataset generated by the Interpro team. Inter-Pro-to-Go mappings are highly reliable but have limited coverage due to the labor-intensive nature of manual curation. We assessed the correspondence between domain Go-term pairs and found that our predictions successfully retrieved 737 out of the nearly 6,000 inter-Pro-to-Go associations. Moreover, many of the associations obtained by domain-to-Go involved closely related Go-terms on the gene ontology directed acyclic graph. For instance, in inter-Pro-to-Go, there is an association between the histone acetylation Go-term and the nuclear receptor coactivator domain, which was one of the most frequently associated domains in the manual associations. We identified the association between this domain and the Go-term histone H2B acetylation, which is a child term directly related to the manually associated histone acetylation. This comparison demonstrated that the proposed method was able to retrieve many of the mappings in the inter-Pro-to-Go set and provide more specific domain function annotations. Here we can see that randomly selected pairs with high S-score are commonly present in the manually curated inter-Pro-to-Go mappings. To assess the biological significance of our findings, we examined 3 specific domain-to-Go mappings. Let's focus on one of these mappings: the association between the domain helix hairpin helix DNA binding and gene ontology biological process term DNA ligation involved in DNA repair. These mappings demonstrated the role of this domain in repair processes, a connection supported by the manual associations in the inter-Pro-to-Go between this domain and the biological process term DNA repair. The domain-to-Go mapped Go term is positioned as the child term of the manually associated term, further reinforcing their relationships. Additionally, there are articles available that explain the biological association between the predicted Go term and this specific domain. This example serves as a clear illustration of how domain-to-Go successfully associates domains with biologically relevant Go terms. We wanted to explore the protein function prediction capabilities of domain-to-Go, although its primary focus is on domain function prediction. Using the CAFA3 challenge benchmark dataset, we compared domain-to-Go's performance against 3 methods: 2 CAFA baselines, BLAST and naive methods, and the manually curated inter-Pro-to-Go annotations. We tested 2 individual protein function prediction methods within domain-to-Go, referred as domain-to-Go-S and domain-to-Go-E. The calculated co-occurrence similarity and evidence scores were used as probabilistic prediction scores for protein Go association predicts in these methods respectively. Coverage on the benchmark protein set for these methods can be seen at the top of the bars. As domain-to-Go predicts protein functions based solely on domain information, it was expected that both domain-to-Go-S and domain-to-Go-E would exhibit lower coverage values compared to the baseline methods. The top line plots show the Fmax performance of the methods, with lighter bars representing full mode evaluation results and the darker bars representing partial mode evaluation results, focusing only on proteins with predictions. Both domain-to-Go-S and domain-to-Go-E outperformed inter-Pro-to-Go and the baseline predictors in predicting molecular function and biological process terms. However, in predicting cellular component terms, domain-to-Go-S, domain-to-Go-E, and inter-Pro-to-Go models performed worse compared to the baseline methods. The relatively low predictive performance of domain-to-Go and inter-Pro-to-Go in cellular component category may be attributed to the challenge of associating domains with cellular structures, as domains can be part of various proteins functioning in different locations. Additionally, we developed a hybrid protein function prediction approach that combines sequence and domain-based data. The domain-to-Go BLAST prediction set was created by deducing a consensus from domain-to-Go-S and BLAST prediction sets. The CAFA3 benchmark analysis on cellular components predictions using domain-to-Go BLAST resulted in a significant improvement over domain-to-Go-S and slightly better performance compared to the BLAST. This highlights the benefit of combining different data sources and utilizing a larger knowledge space. The second line plots display the estimate performance of this method, providing insights into their ability to make accurate and specific predictions, as this measure depends on the information content of the predicted Go terms. In the partial evaluation mode, domain-to-Go outperformed the baseline predictors for all Go categories. The domain Go term mappings generated in this study have the potential to facilitate large-scale Go annotations for unknown proteins. The scalability is a significant advantage, which is a critical issue for most of the state-of-art protein function prediction methods. In our study, we exemplified the scalability by utilizing the final domain-to-go mappings to predict GO-term annotations for over 500,000 proteins in the UniProt-KB SwissProt database. We achieved this by propagating the domain-associated GO terms to the proteins that contain those domains, leveraging the protein domain annotations provided in the InterPro database. The resulting protein function prediction set encompasses over 5 million predictions, covering nearly 300,000 proteins. Notably, this entire process took only 4 minutes to complete on a regular consumer-level computer. Domain-to-Go offers not only the advantage of speed but also interpretability, unlike black box models, since its function predictions are localized to specific regions, structural units in proteins. Furthermore, the methodology of Domain-to-Go can be readily applied to predict diverse biomolecular relationships, including protein associations with diseases, phenotypes, and drugs. If you are interested in exploring further, we have made the source code, datasets, and the study results fully available on our GitHub repository. We also recommend exploring our preprint publication for a comprehensive understanding and additional insights. Finally, I would like to thank our group, particularly our advisor Dr. Doğan, for their support. Thank you for your attention, and please feel free to ask any questions you may have.